that was interesting. Yeah, so I was a graduate student, then switched to a postdoc here at Rockefeller in the same lab, in Fernando Nottebohm's lab. And I remember at that time when there was a big interest in trying to understand the molecular biology of learning and memory. And myself, Fernando, many other people had different ideas of what you needed to do. And I was influenced to do some kind of play song and shock train the animals, to avoid shock when they hear a song. And so classical, Pavlovian conditioning kind of experiments, but just do it in songbirds instead of mice. And accidentally, we found that some of these birds we couldn't get gene activation in the song learning nuclei to understand their functions with this classical conditioning. It didn't happen. And we knew these genes were responsive to electrical activity in the brain. So we knew something's got to be going on, but why aren't they activated in these song learning nuclei? So we thought maybe after the bird is a certain age, nothing's going on. They're at least at the molecular level. And then accidentally, we came across some birds that had some gene expression in their song nuclei, these activity-dependent genes, and wondering what in the world is going on. And that I realized before I put them in the shock training experiment, right, those birds were singing in the other room adjacent to me, before moving them into the classical, the conditioning apparatus. And this, and we did more experiments to prove, okay, this is singing-driven gene expression, vocalizing-driven gene expression. And that was a key that switched in my mind. Look, try to do experiments that focus around the animal's natural behavior, what it has evolved to do, instead of forcing our human-derived anthropomorphic views onto these animals. Right. And ever since I started thinking that way, it's been one discovery after another.